I'm Sophia, and today I'm going to talk about DNA replication. So here we have these two DNA molecules, and how do they replicate? They replicate in the same way, and they replicate by one particular model. And today we're going to figure out what that model is. So first, how do those DNA molecules replicate? So for example, if we hold this hand, then we're copying all the nucleotides on this hand and placing them into another hand. And then what we do is that we have this very first strand with the old and the new, and then we have this strand with the old and the new. You might have already guessed the model of DNA replication, but just in case if you don't know it, we'll actually talk about it here. So here we have these two molecules. They have to reproduce their nucleotides. The only thing that we really know is that its DNA is always a double helix. It's always a double helix, so that's why its DNA lends itself to a very good method of replication. So what's happening is that you take this double helix and split it apart. So here in this figure of Campbell biology, you're noticing how it replicates. Okay, so here you have this strand, nucleotides, and then the two are separated, and then the dark blue strand are connected to form the sugar phosphate backbones of the new light blue strands. And then here we have the conservative, the semi-conservative, and the dispersive model. So first you have this parent cell containing this parent DNA. And in the first replication, there's going to be one new, one old. And then out of the new, there's all new. And then out of the old, there's one new, one old. And that repeats. And then during the semi-conservative model, we have this parent cell, parent DNA. First replication, half new, half old, half new, half old, half new, half old, new, half new, half old. And then here in this dispersive model, you have it a little bit different. Then you have this parent molecule, parent DNA, and then we have a little bit new and old, 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 new and old. So it's all mixed up. In the semi-conservative model, it's super clear. In the conservative model, it restores the parental double helix. But what is that actual model? You might think it's conservative, but it actually is not. Because you might think it's quite simple reproduction model, but what happens is that you need to have some new DNA, some mutations, because otherwise there might be two copies of the mutation, and there might be difficulties because they might not have the same alleles and all those troubles, because maybe they're from the different DNA strand. So that's a lot of difficulties. That's why it is not conservative. So first, Matthew Michelson and Franklin Stahl, they cultured the Escherichia coli. And then, for several generations, it contains this heavy isotope of nitrogen, 15 nitrogen. And then it recultured, and that 14 nitrogen is a very light isotope. And then they took one sample after the first, and one sample after the second replication. So they extracted that DNA, and then they centrifuged the DNA to separate the DNA of different densities. And then during the first replication, see, this is what happened, and in the second replication, it's already split into what's less and what's more dense. And then here during the conservative model, we didn't have this during the first replication, so it could not be the conservative model. What about the dispersive model? So during the dispersive model, we had this first replication that was exactly the same. But how could you even stronger have the strong idea that it's definitely not the conservative model? You need more ideas. And why is it not that dispersive model? Because the old and new, you're just synthesizing the DNA fragments from here and there. It isn't so difficult. Well, what is happening is that it actually spreads, and then you notice that it isn't because it's again the second replication, and then the dispersive, it still stayed all mixed up. While in the semi-conservative model, it already got split. So you have this, and they didn't get mixed up like this during the first replication. Semi-conservative, first replication, dispersive. Second replication, dispersive, and second replication, semi-conservative. You already recognize what's different. So here, there's the origin of replication. So again, in one of those DNA molecules like this, there's a bunch of origins of replication. And then you could actually observe the origins here. So first you have one origin, let's say here, or actually you could have this a little bit better DNA molecule, and then this. So now you could have, let's say, an original replication here, and then you could, let's say, have an original replication here. And then what would happen is that you could make a bubble here, make a bubble here, and then those bubbles will reach here, and then they'll merge. Two bubbles will apparently all the time they'll merge. And then we have this parental and the new strand. And then what happens, you have this many sites along this giant molecule basis. This is only a fragment of this giant DNA molecule. It's a very tiny fragment of that giant DNA molecule. And this actually happens during S phase of interphase and mitosis and meiosis. And those are just two types of cell divisions. And then what is happening here? So here we have a bunch of enzymes. So let's say the replication proceeded until here, and then we have this topoisomerase that acts the winding fork. Sorry, you winded here, and then it relieves the strain caused by unwinding. So it isn't like so much strain. And then there's this helicase that reopens, opens. So the topoisomerase is here, and the helicase open, and then opens this, and then opens this. So you got more and more replication, and then the topoisomerase just moves so that it won't just relieve the strain caused by unwinding. And then you still have to stabilize these little areas, and then there's those single strand binding proteins that stabilize those unwound parental strands. They stabilize them so that they won't be unstable, like topoisomerase is here, and these like all spread. And then the primase, it synthesizes RNA primers. And that occurs ahead of DNA polymerase 3 in every strand. And there's also another type of DNA polymerase, which is called DNA polymerase 1. First, how does it produce one more nucleotide? So first we have those 3 phosphates, 1 phosphate, 2nd phosphate, 3rd phosphate group. And then we have that template strand, which has that nucleotide, and we need to add an extra. So the DNA polymerase releases that nucleotide and the pyrophosphate. And that pyrophosphate can react into 2 inorganic phosphates. And that's almost the same as what occurs when you hydrolyze 2 ATPs. What happens is that they release 2 inorganic phosphates and 2 ADPs. Same thing that occurs in glycolysis of cellular respiration. So what you have here is you have this lagging and leading strand synthesis, because there's 2 strands. And then first you're going to look at the leading strand. So first, this RNA primer is made, and the DNA polymerase 3 starts to synthesize the leading strand. And it is elongated continuously as that helicase, it opens the fork. And then the primase, it joins the RNA nucleotides into that first primer for that lagging strand. So it makes that RNA primer. And the DNA polymerase 3 adds DNA nucleotides that forms this Okazaki fragments 1. And an Okazaki fragment in a prokaryote is 1,000 to 2,000 nucleotides long, while an Okazaki fragment in eukaryote is actually only 100 to 200 nucleotides long. After reaching that next RNA primer, the DNA polymerase 3 detaches. Done. And then the fragment 2 is primed. So it's just the RNA primase passes through, and that's that new primer. And the DNA polymerase 3, it adds some DNA, and then it detaches when it reaches the fragment 1 primer. And that DNA polymerase 3 replaces the RNA, which occurs in transcription, which is a completely different process, with a DNA, which occurs in DNA replication, which adds nucleotides to the 3' end and the 3' end of fragment 1, and then it later adds the nucleotides to the 3' end of fragment 2. And that's where that DNA polymerase 1 participates. Shoot! And then DNA polymerase 3, and then DNA polymerase 1 finally goes down. Now the DNA ligase, it forms that little bond between that new DNA and the DNA of fragment 1. So there's just a little blip, blip. The DNA ligase helps make sure all the DNA is down. But there's some important idea that some of that DNA gets lost. And in some cases, you could at some point actually lose functional genes. Oh no, how does that occur? Well, that occurs because of the shortening. So we have this last fragment and the next two last fragments. So we remove primase and replace with DNA where that 3' end is available, which leaves most of it down, but that last RNA primer can't be turned into DNA. And then after the second round of replication, we have a different leading strand and a different lagging strand. And then after further rounds of replication, we have shorter and shorter and shorter and shorter daughter molecules. So it becomes short, and then it could lose functional genes. And actually, prokaryotes don't have a way to deal with this, but eukaryotes have those repetitive and non-coding sequences called telomeres at the end of the DNA, and they're stained orange in the mouse chromosomes. So what happens here is that they have undergone many cell divisions. So what happens is that it contains this RNA molecule that's called a functional RNA, so it doesn't get translated, and it uses this template to extend the leading strand, preventing the shortening. And then it allows the lagging strand to maintain a given length. And we have this enzyme called telomerase that does that, which helps the telomeres. And telomerase is not normally active in most human somatic cells, but that activity varies from one tissue to another tissue. And then we have a very large activity of telomerase in the germ cells, which means that the children would be more likely to be healthy because it results in telomeres of maximum length in the zygote. Thank you.